Howdy there, folks, and welcome back. So, for this lecture, we are going to dive into the wonderful world of the human microbiome. And this is a very complex part of our bodies, and I can't do the whole thing true justice in just one lecture, but we're going to take a kind of a superficial overview of the human microbiome. Remember, within a body of a healthy adult, microbial cells are estimated to outnumber human cells 10 to 1. Actually, that is a little bit of a microbiology urban legend. The closer count is about a 1-to-1 ratio. And interestingly enough, every time we take a healthy number 2, it skews the numbers in the direction of our human cells rather than the microbiome. But the vast majority of microbial species in the human body have not yet been analyzed because their growth is dependent on a very specific microenvironment in the human body, and we can't reproduce that in the lab yet. So we're still not 100% sure about all the ins and outs of our microbiome. We've evolved in the context of complex interactions with complex communities, both on us and within us. Microbes play an important part of an organism's phenotype beyond just symbiosis. And remember, from genetics, phenotype is the physical expression of an organism's genes. We can't separate our genes from the context of our microbes because they are so interlinked. Microbes have been shown to behave differently when together. Planktonic or free microbes tend to have completely different phenotypes and behavioral patterns than ones living in a biofilm that are living and working together, almost like a multicellular organism. Microbiomes provide a wide range of metabolic functions that humans lack, and DNA-based studies are happening worldwide to identify and understand the functions of the human microbiome and how our bodies interact with them and each other. The human microbiome is unique to each individual. Although we may have microflora in common, they differ in their locations and their coverage on the body. Even the right and left sides of your body can be vastly different from each other in the context of your microbiome, which is kind of cool to think about. Studies contrasting the gut microbiomes from different countries have identified systematic differences in microbial compositions. Smaller international studies have also identified geography as one of the major sources of large-scale variation in the human microbiome. Among possible drivers for this variation, diet has been suggested as an important contributor, along with other factors including geography, early life exposures, and genetics. Here in the Bay Area, it's really interesting. We have vastly different microclimates depending on where you live in the Bay Area. Even within just going to San Francisco, the city itself has vastly different microclimates. So the interesting thing is that folks that live and work in a particular area may have vastly different microbiomes than folks that live in other areas. Case in point, in New York, they did this really cool study where they swabbed the trains, some brave individuals there, but they swabbed the trains and they actually found that the microbial composition was different for every borough in New York City, which makes sense if you think about it, because New York City is still very divided according to socioeconomic lines and also ethnicities, and people of certain ethnicities tend to eat certain foods that maybe folks from other ethnicities that live in a different part of the city don't. So diet and location again have played this crucial role in kind of dividing up where the lines are for microbiomes on folks living in a large city. I would be really interested to see somebody do a similar study with folks in the Bay Area. So I have a feeling that it's going to be very similar. Depending on where you live in the Bay Area, you're going to have a completely different microbiome. Although some recent studies suggest that birth method plays less of a role in the establishment of our microbiome than we previously suspected, researchers feel that babies born via cesarean section have an overabundance of opportunistic species like Haemophilus, Enterobacter, Veillonella, and Staphylococcus. Babies that are born via the vaginal birth canal usually get coated in mom's microbiome, and it gives them kind of a boost in the right direction, so to speak. Other hallmarks of health from the microbial ecology perspective are the ability to resist perturbation or disturbance and to return to a healthy state afterwards. This might result from the entry of a pathogen, alteration in diet, medications, moving around to different locations, going swimming in the ocean, taking a bath. All kinds of different things can cause these disturbances, and whether or not resistance, the resistance is the ability for the microbes to not get washed off or kind of stick tight where they are. And resilience is, can they come back after a disturbance? Like a planet's biosphere, you can think of yourself as a patchwork of many different ecosystems. You have wet areas, dry areas, oily areas, areas of different pH, and areas of different temperatures. We're going to take a tour of the microbes that are found normally in certain parts of the body. This is by no means an exhaustive list, and I don't want you like pummeling your brain to try and remember all the genus and species names for these microbes. I want you to have kind of an overall picture of, what the body is like, patchwork style. So with the scalp, it's got a distinctive skin area with relatively high follicular density and sebum production and secretion. So it's got a lot of hair and a lot of oil. When hair is present, it's a relatively protected area with microflora that are often unique to the rest of the body. When compared to skin and other body areas, the scalp skin is relatively thicker, it contains more blood vessels, so it's going to be warmer, and it presents an environment that's rich with sebum and dense with sebaceous glands. It inhabits unique combinations of biota domination. Common microbes of a healthy scalp include Malassezia globiosa, Malassezia restricta, Propionium bacterium acnes, and Staphylococcus epidermidis. So moving on to the nose is a primary defender against inhaled pathogens because of the cilia and the mucus lining inside trapping inhaled microbes. Inflammation from viral infections and allergic reactions can occur, and often these are treated with inhaled medications and oral antibiotics. These can vastly shift your microbiome in the nose. There's a delicate balance of microbes that are maintained to keep that environment very healthy. Weakened immune systems can throw off that balance and allow the wrong microbes to grow out of control. Now, there's often I don't know if you want to say a controversy, but it's been postulated that eating your boogers might actually be good for you because it helps train your immune system. This came up in a class that I was teaching several years ago, and I didn't know if it made intuitive sense. Like if you're eating microbes that have been trapped in your nose, then it's going to introduce it to your body. So could it possibly help? So that made me do a little bit of internet digging, and I actually found this video that we're going to watch here about picking your nose and eating it. And I hope you enjoy this as much as I do. All right, here we go.
Julia, we're rolling.
Ah, God! Hi, my little nuggets. Julian here for D-News. Picking your nose. You all do it. I know you do. Don't lie to me. But once you've mined your golden gobs, do you wipe it on tissue? Do you pick it and flick it? Do you eat it? The behavior is not uncommon in kids at all, and a lot of adults still do it. Usually when no one's looking, but they do occasionally get caught on the Jumbotron at a sports game. But is there a reason to be ashamed of it? Or worse, could it actually be unhealthy? Well, the social stigma makes sense. Mucus is inherently gross. It's stringy and sticky and drips out of you when you're sick. Once you learn the purpose of mucus, it adds another layer to the yuck pile. Mucus's job is to snag dirt, pollen, and pathogens. It's actually 95% water and 3% mucin, which are lubricating gel-like proteins that hold a lot of water and have a high molecular weight. The last 2% is assorted particles ranging from antibodies to fungus balls. So knowing that one job of your nose goop is basically to catch the nastiness in the air before it makes it to your lungs, why, oh why, would anyone put a booger in their mouth? Well, because they taste good. So I have heard. Associate Professor of Biochemistry at the University of Saskatchewan, Scott Napper, thinks so too, and moreover, thinks boogers are sweet as a way of enticing us to eat them. Since they've got weakened pathogens in them, perhaps eating them helps our immune system learn to fight whatever might be in the air around us. I'm gonna call shenanigans right there though, because you're already swallowing a ton of mucus without knowing it. You make about 1 liter to 1.5 liters of mucus daily, and much of that gets pushed back by cilia and flows down the back of your throat into your stomach without you even realizing it. You're basically swallowing oodles of snot constantly, so a dried boogie that you put there manually isn't going to help you out very much. Instead, I would like to offer a different hypothesis on why boogers are allegedly sweet. Mucus also helps with your sense of smell, cutting up molecules that enter your nose and delivering them to receptors in your nasal cavity. A 2007 study claimed that the delay in the time it takes different pieces of the molecules to reach receptors actually helps us differentiate smells, and this was backed up in a 2010 study where mice with inactive mucus lost the ability to identify a target smell. When it's dry out, the mucus dries up and can't transport those molecules well. Mucus also keeps the tissue underneath it from drying out and cracking, keeping the blood vessels in your nose safe from infection. So, in order for mucus to do all its jobs of lubrication and protection and smell reception, the mucin needs to stay full of water. And how does mucin trap water? By being coated in hundreds of oligosaccharides. That's right, sugars. Boogers aren't sweet because you're supposed to eat them. Boogers are sweet because they've got a job to do, and their taste is a byproduct of their strategy to do it. So it looks like eating your goobers isn't beneficial, but it's not harmful either. Just don't pick so much you scratch up the inside of your nose and cause bleedings and infections. And probably avoid doing it on a date. But aside from that, science has no criticism if you pick and lick. So, boogers are gross, but apparently, according to a science professor, they taste good. But you know what doesn't? Earwax. What's that stuff for? Trace tickles your earwax. Here.
It's mostly dead skin cells, proteins, and oils added by the apocrine gland. The junk is there to protect the inner ear from water or infection.
Given that there's really no pro or con to a tasty nose feast, do you still think it's gross? Probably, but let us know in the comments anyway, or check out our new Reddit page, r/dnews. Subscribe for more tasty treats, and I'll see you next time on D News.
So, I hope that was as illuminating for you as it was for me, and that you enjoyed that little nugget. So, in the nose, you typically will find the bacteria Staphylococcus epidermidis, which we play with in lab, and these bacteria form a biofilm that coat the internal mucosal lining. Staphylococcus aureus, when it's found in humans, is usually found in your nasal areas or around your ears. And it's fine when it's kept under control by protease found in Staph epidermidis, but if it's left to grow out of control, Staph aureus can become pathogenic and cause infection. Aspergillus fungal spores are often inhaled through the nose as well. If the immune system fails to clear these, the mold can grow in the lungs and cause you to be very, very sick and even die in some cases. Corynebacterium accumbens is a bacteria that's rarely a pathogen, but if it enters the bloodstream from a torn blood vessel, it can cause serious infections. Another reason why it's probably not a good idea to pick your nose in case you introduce bacteria where they're not supposed to be. The oral cavity of your face hole has a wide variety of microbes that are normally found there. A wide variety of microbes regularly enter this oral cavity, and brushing and flossing clears some of the built-up biofilm, but by no means all. Saliva, pH, temperature, and the immune system all prevent many species from surviving or coating the entire mouth. Oral antibiotics will also inhibit growth of these as well. Symbiosis of the oral microbes that are able to survive these conditions form an elaborate scaffolding that lives on the tooth enamel and at the interface with the gums. It forms a barrier for incoming bacteria, and genetic disorders can lead to the oral microbiome weakening. When I was teaching at City College in San Francisco, UCSF was doing this really cool study for several years, and I need to reach out to them and see where they got with that. But they were paying students to participate in a study where they were consistently swabbing their mouths, looking at their microbiome. And what they were trying to do is compare the microbiomes in the mouths of healthy folks to people that had a condition called dry mouth. And people with dry mouth tend to have worse cavity issues than people that don't have dry mouth. So they were investigating the linkage and seeing if there was perhaps a bacterial difference in folks that have normal lubrication with saliva in their mouth going on and folks that have these drier conditions. The oral cavity is a wide variety of microbes that are normally found there. Fusobacterium species, the bacteria is a larger bacteria that helps form scaffolding that I was talking about for many other bacteria in the oral biofilm to stick to. Streptococcus mitis bacteria typically forms a biofilm on the hard enamel surfaces of the teeth, and if the gums get inflamed, it can enter the bloodstream and cause infection. Prevotella species of bacteria have natural antibiotic resistance genes, and they can attach to the epithelial cells or other bacteria and cause larger infections in inflamed areas. And then our great friend Candida albicans, which causes a host of different fungal infections in humans. This fungus can cause an oral infection known as thrush. On your skin, there are several different skin environments: oily, dry, moist. Some microbes prefer one over the other, and the skin's got natural defenses including slightly acidic sweat and antimicrobial peptides. Microbes hide in the crevices to recolonize the skin after washing with soap. Antibiotic washes and oral antibiotics disturb the normal balance of the microbes on the skin, and they can actually cause you more problems than just letting those germy germs hang out where they're supposed to. There's a normal balance of microbes on the skin that protect introduced microbes from harming us. Damaged skin, though, gives opportunities for microbes to invade the bloodstream and cause serious illness inside us. Propionum bacterium acnes is a bacteria that colonizes healthy pores, but if the pores become clogged, it grows out of control and it can cause pimples. This is the primary bacteria that causes the condition acne vulgaris, which is what most teenagers go through while their body chemistry is changing. One of the more interesting studies that I've seen lately is when they were looking at acne and strain pathogenicity in this Propionum bacteria acnes species. And they found some interesting things. They found that folks that have more serious acne problems typically have a strain of this bacteria that's different than folks that don't have acne problems. So what they're trying to figure out how to do is come up with some type of cream that folks that have acne vulgaris can put on their bodies to help reintroduce the good strain of bacteria and wipe out the bad strain of bacteria, which is kind of cool if you think about it. Staphylococcus epidermidis is a bacteria that normally colonizes on the skin, but when it clogs pores, it can grow out of control and infect the pores, and you can get ingrown hair issues and also pimples. Staph aureus bacteria can also infect clogged pores, like Staph epidermidis does, and even worse, many antibiotic-resistant strains of Staph aureus make it very difficult to treat the infection. Trichophyton and Microsporum fungi feast on keratin in the skin and cause the condition that was known as ringworm. This is actually a fungal infection and not a true worm, so a little interesting bit of trivia there. Some other skin hot spots include the armpits, the genital area, and the feet. They mostly stay moist and warm and can harbor other types of microbes. So what's the big deal about this? There's actually been a skin microbiome project that was going on to look at different folks' microbiome and see if there's some way that we can develop new products like clothing that don't have to be washed as often because they work with our microbiome. So I've got this short video that I wanted to show you that kind of has to deal with that.
Hello, I'm Tim McGee from LecoLab, and you are human. But it turns out most of the cells in and on your body are not. In fact, if we were to zoom in on your skin, we would find an entire thriving ecosystem of bacteria, fungi and archaea. We call this the skin's microbiome. We still don't know a lot about it yet, but we do know that it's important. Scientists suspect it's linked to our immune system development, wound healing, disease protection, acne, psoriasis, and a lot more. The ecology of our skin's microbiome can have a dramatic impact on our own health and well-being. Yet the products we use on our skin have never even considered our microbiome. We want to change that. By bringing together rigorous science and design, we hope to understand how our microbial life responds to the products we use, and how this might impact our well-being. As biologists, we want to know how different oils or textiles change our microbial ecology. As designers, we care about how products work with the life that is on us, in us, and around us. Let's take a moment to imagine what our products might be like after we understand our own microbiome. What if soaps, lotions, and makeup, instead of killing your natural microbial ecology, made it more diverse, more personalized? What if your cell phone could monitor key microbes on your body and let you know when they were having trouble and needed a boost? Imagine if our jacket could keep diseases from spreading on the bus or subway by using a diverse set of microbes to fend off the invaders. Or what if our jeans didn't need to be washed because the microbes kept them smelling fresh and clean? These are a few of the scenarios that we hope to make a reality. For this project on Experiment.com, Sean Gibbons and I joined forces. Sean is a current PhD candidate in the biophysical sciences at the University of Chicago. To kick off our work, we will collaborate with volunteers who will change their grooming habits and try out new ways to wash their skin. Each volunteer will receive our Microbiome Skin Kit. That includes swabs, sample tubes, new grooming lotions or soaps, as well as some early prototypes of our special cleaning products. We will also be observing them and asking questions about their grooming behaviors and products to see how well people adjust to these new ideas about washing. The volunteers then swab several areas of their body several times a month and send those samples off to the laboratory, where the DNA of the microbes is extracted and then sequenced. Sean and I get the results and figure out which populations of microbes are changing as the volunteers change their behaviors. The data from both the DNA study and the behavioral study gets analyzed, synthesized, and turned into a useful scientific and design tool. It can help us and others refine new product development concepts, such as novel washing products that can respond to our microbiome. We will be able to pave the way for new design projects to use similar analysis tools. And the research itself can lead to new publications about how the skin's microbiome supports our own health. In short, we can do amazing things.
So the rest of this is a come all ye speech, kind of a pitch, so I don't want to bombard you with advertisements. But really cool, right? So if you think about this is kind of a brand new world for business and science to kind of hook up and do some pretty amazing stuff. So, your gut. Your gut contains the most extensive and diverse habitat in or on your body. It's not easy for microbes to get there either. They have to run the gauntlet to establish themselves. Some of the major barriers for microbes entering the gut are a low pH, saliva and bile, immune system, finding a place to attach in the intestinal wall, and surviving a widely varied diet. The gut microbiome consists of microorganisms that live in the digestive tracts. It's the largest reservoir of human flora that we're aware of, and it's estimated to have 100 times as many genes as there are in the human genome. 300 and 1,000 different species of bacteria live in our guts, and fungi and protozoa make up part of this microbiome as well. Commensal or non-harmful bacteria can also form a mutualistic relationship. Microbes perform a host of useful functions such as fermenting unused energy, training the immune system, preventing growth of pathogenic bacteria, regulating the development of the gut bacterial microbiome, producing vitamins such as biotin and vitamin K, and producing hormones to direct the host to store fats. Probably the body to store fats would be my guess. So these are all beneficial things that our gut microbiome can do for us. The gut microflora is mainly comprised of 3 enterotypes, which are necessary for the digestion of carbohydrates, animal proteins, and fats in humans. They will vary depending on diet, and as your diet changes, their percentages will change as well. Now, just to give you a little bit of another interesting bit of information, some of us, if we have like a piss poor diet and we don't eat healthy foods, which we'll talk about later on when we talk about the differences between the good foods to eat and the bad foods to eat with respect to our microbiome. Some folks that have a bad diet will actually be colonized by bacteria that exert this crazy type of mind control, and they actually can secrete chemical hormones that affect your brain and make you crave more of those fatty, bad for you foods. So in some cases, we are actually at the mercy of our gut microbiome when it comes to certain food cravings that we have, which is really crazy if you think about it. For the microbes that manage to colonize the gut flora perform regular tasks of digestion, vitamin production, and many others. Gene transfers between a myriad of different species in the gut can generate new combinations of drug-resistant superbugs like Clostridium difficile, which is very common in nursing homes and step-down units and hospitals. It is very difficult to treat. It is also one of the smelliest infections you're ever going to experience if and when you become a health professional. You will know that smell. So I have a TED Talk here about mind-altering microbes and how the microbiome can affect the brain and behavior, which I just find fascinating. So I hope you enjoy this TED Talk.
So it never ceases to amaze me that each of us carries around a 3-pound mass of cells in our heads that controls literally everything we do. Importantly, though, the brain doesn't act in isolation from the body, but rather it responds to the needs and experiences of each of our organ systems. Now, here's a staggering statistic that some of you may have heard before. Our bodies are comprised of 10 times more microbial cells than our own human eukaryotic cells. These microbes, which are primarily bacteria, but also viruses and protozoa, they're part of our normal flora, and they make up what's called the commensal microbiome. In the intestines, there are 100 trillion of these bugs, reflecting over 10,000 unique species and contributing 150 times more genes than our own human genomes. It's even estimated that collectively, these microbes would weigh 2 to 6 pounds, which is up to twice the weight of an average adult human brain. More and more, we're learning that these commensal microbes that make up us have co-evolved to play fundamental roles in normal brain development and function. So we can study the role of commensal microbes by raising mice as completely germ-free and recolonizing them with whichever microbes are of interest. And by these types of studies, we're learning that commensal microbes regulate several complex behaviors, like anxiety, learning and memory, appetite and satiety, among lots of other behaviors. So you can see now that by studying this microbe-brain interaction, we can learn really important lessons about how microbes can contribute or affect our brain health and disease. So you might be wondering, how in the world does a microbe that lives in your gut affect your brain? And there are many different mechanisms. One way is by activating the vagus nerve. So the vagus nerve contacts the gut lining and extends all the way up to the brainstem itself. And this is a mechanism by which the bacterium called Lactobacillus rhamnosus affects depressive-like behavior in mice. So in a task that measures depression-related despair, mice that have been treated with this bug exhibit less depression-like symptoms. And this is not seen if the vagus nerve is severed. Another way by which microbes can affect the brain is by activation of the immune system. So about 80% of the body's immune cells reside in the gut, and immune abnormalities contribute to several neurological disorders. And this is one mechanism by which the bacterium Bacteroides fragilis prevents a mouse version of multiple sclerosis. So mice that have been treated with this bug are more resistant to the disease, as shown by the red line in this graph. And this depends on the activity of a special subset of immune cells called regulatory T cells that expresses the marker CD25. So if we block the activity of this immune cell, then the beneficial effects of the bugs are prevented. Another way by which bugs can affect the brain is by activating the gut endocrine system. So gut endocrine cells are primary producers of neuropeptides and neurotransmitters. Gut microbes themselves can also produce metabolites that could affect brain function. And this is one pathway that we think is involved in the microbe-based treatment that we in the Patterson and Masmanian labs here at Caltech have used to treat autism-like symptoms in mice. So by treating mice with this bacterium, Bacteroides fragilis, we're able to correct core abnormalities, such as the communication deficit that's depicted here. That's a hallmark symptom or diagnostic symptom of autism. So here, mice that are autistic-like display less communication, as depicted by the blue bar, and treating them with the bug corrects this effect, as shown by the red bar. So I think that the implications of these discoveries is huge, because what if we could, without a single invasive procedure, treat disorders like autism, depression, and multiple sclerosis? Microbe-based therapeutics might offer a way to build a stable community structure that can impart long-lasting effects without the need for a continuous treatment. Also, since microbes are relatively easy to manipulate and even eliminate, they can be readily modified for better functioning, regulatory control, targeting, and even delivery. So as a take-home message, I want you to remember that not only are we made up of mainly microbial cells, but that some of these cells can be truly mind-altering, affecting our brain development, function, and even our behavior. And also, in light of several studies showing important roles of commensal microbes in a variety of biological processes, from nutrition and immunity to now brain and behavior, consider all the things that we do on a day-to-day basis to change or disrupt our microbiome, and how this might affect our health and predisposition to disease. Thank you.
So some fascinating stuff to think about with regards to our relationship with the bacteria that live in us and on us. In the gut, we also have some extremophiles, Bacteroides, so thetoiodomicron, there we go. Bacteria ferment simple carbohydrates in the gut, releasing hydrogen and carbon dioxide. Methanobrevibacter smithii is an archaeobacteria that consumes hydrogen gas from Bacteroides and produces methane, which is lost from our gut as, you guessed it, farts, gas. Ruminococcus species of bacteria can be found in significantly high numbers in the gut flora, and they break down cellulose in the gut, helping with digestion. Helicobacter pylori bacteria has a helical shape and colonizes the stomach and the upper GI tract. It's been known to be a major cause of stomach ulcers, although with many Helicobacter pylori infections, folks that have them don't get ulcers. There's actually a really nifty story behind Helicobacter pylori. It was determined that a strain of pylori was causing gastric ulcers. And so the scientists that discovered this figured this out and figured out a way to treat these infections so that they would go away. But when he was later studying indigenous cultures in South and Central America, he found that humans have Helicobacter pylori as part of their normal microflora, and that in a lot of those folks, it never caused ulcers. So what's the deal? Well, it turns out what they think happened is that when we started bombarding our bodies at a very young age with different antibiotics, we killed off the good strains of Helicobacter pylori that were living commensally with us and allowed the strain that does cause ulcers to thrive. And this caused a shift in our microbiota. And now if we get introduced to these Helicobacter pylori, 9 times out of 10, especially in our civilization here in the United States, North America, you're going to get the one that causes ulcers more often than not. So it's problematic that way, but it's also really neat to see that we can affect change, both good and bad, in our gut microflora. The urinary system is almost sterile due to urea and other chemicals, and urine often flushes out microbes that find their way in. Anytime you bypass a mucosal membrane, though, like introducing a catheter into the urethra, you can introduce microbes directly to the bladder, where a biofilm can grow and cause a bladder infection. The vagina has a low pH due to lactobacillus secreting lactic acid and hydrogen peroxide. Lactobacillus normally maintain a low pH, while other species are kept in small numbers in the vagina. If lactobacillus decreases from antibiotics, Candida albicans can take over and cause a yeast infection, and Gardnerella vaginalis can grow too much and cause bacterial vaginosis. E. coli from the GI tract can infect the urinary tract due to poor hygiene and contamination from the nearby GI tract opening. So there is a project called the Human Microbiome Project, and their goal was to map the human genome initially, and now they're trying to map the microbial genome that live on humans, which is very interesting. And I've got a short video here to show you about this.
My name is Curtis Huttenauer. I am an assistant professor in the Department of Biostatistics at the Harvard School of Public Health, and I work on the Human Microbiome, which is the collection of microbes, single-celled organisms like bacteria, viruses, fungi, and many other organisms that we carry in and on our bodies. There are about 10 times as many microbial cells in our body as human cells. Our skin, our mouth, our airways, every exposed surface in the body carries its own naturally occurring microbial community, which typically has evolved with us to promote health and protect the human body in that particular environment. Our group helped with one of the main consortium analysis efforts in order to interpret these sequences as a map of what our normal microbial variation is. So this really gives us a definition of the boundaries for over a dozen different microbial habitats associated with the human body, of what kinds of microbial communities can be normal in health. The microbes associated with our bodies give us another way to potentially assess lifelong risk of disease or immediate risk of disease outcome. What we're learning right now is how better to use our microbes to predict the risk of autoimmune or inflammatory disease onset, IBD, diabetes, diseases in that family, and how to predict the risk of their outcome. So for example, the onset of a Crohn's disease flare, using not our DNA, not our own genes, but our microbes and their genes and biological activity. One of the reasons, or one of the areas why a lot of people in this field are very interested and very motivated about the topic right now, is that the microbiome is part of our environment that might help to explain the last 50 years or so very rapid increase in chronic non-genetic disease. If environmental exposures are part of what's causing this increase in obesity, in diabetes, and other chronic conditions, and that's something that is affecting our microbes in a way that can be treated or prevented or used to improve health, that's a huge motivation and something that needs to be worked on now as quickly as possible in order to help ameliorate the rise in chronic disease and obesity. It's a very exciting time now to be working on both computational biology and DNA sequence analysis generally, and the microbiome in particular. These are both very new areas that are changing rapidly. There's a lot of work to be done. I'm looking forward to seeing how the Harvard School of Public Health, our students, our group, and others carry out research in the area.
Pretty cool stuff. So I have one last TED Talk to show you, and this is forward thinking. This is more forward thinking than we've perhaps thought before. And this is listening to this TED Talk, Our Germs, Our Future, and the Human Microbiome as a Community of Self, and how we might be able to view the microbiome going forward and help it help us. From a community standpoint. So here is this last video.
What are we? As humans, what are we really? We humans have been asking that question basically forever, and different cultures around the world at different times have found their own answers to it. Recently, there's been a new answer, that we humans are mostly bacteria. 90 to 99% bacteria. And even cautious microbiologists are observing that this changes how we are able to think about ourselves. And that's interesting because how we think about ourselves changes the assumptions that we make about what health is, what well-being is, what sickness is. It makes new actions available to us for the future. But before we look too far ahead, I've always found it illuminating to look back at different ways that we've answered this in the past. And I want to share with you two very influential ways that we've answered that question, what are we? Maybe we're like a bowl of soup, a bowl of steaming liquids with a dollop of soul on top. 200 years ago, that was more or less what we were, containers for our humors, for fluids of life in balance. They're sensitive to the bad airs around us. When they get thrown out of balance, then we get sick. For thousands of years, this was what we were. But when was the last time you worried about your phlegm being out of balance with your blood? Has anyone here been bled, like medically, with leeches? No, because the majority of us no longer operate in a humoral paradigm. We might say that we're 70% water, but not that we're a homeostatic balance of blood, bile, and phlegm. Maybe we're sealed machines, engines that burn food for energy. Our skin is like our armor, but it's vulnerable in a few chinks. Germs can get in and make us sick. Armies of invisible invaders are coming for us. We're under siege all the time by the germs in our air, in our water, in our feces. And when our feces gets in our water, then it's war. This is the paradigm that we've been living in for about 100 years. It's where germ theory has steered medicine and public health, and it's made possible the huge leaps that we've made in life expectancy and global population. But being an individual human machine has some issues. This mechanistic war on germs has gotten us through epidemics and famines, but it's left us with a whole new set of 21st century health crises. Now, we all know this list. We've all experienced some part of it in our lives. But we have a crisis of mental ill health, of depression, anxiety, stress. We have a crisis of metabolic disorders and the caustic coexistence of hunger and obesity. We have a crisis of autoimmune disorders. The rates of diabetes to asthma are just through the roof. And unlike epidemics of centuries past, these don't kill equally. We have a crisis of massive health disparities. And it's not just in our bodies. The planet is in a climate crisis, which accelerates all of these challenges to our well-being. None of us are fully equipped to meet these crises. We can take pills and go on diets and recycle, but fundamentally, the individual isn't the most effective unit of change. Unless we change what we think we are. Unless we accept that we're not machines, and that war is not the right action, or even the right metaphor to get us through the coming decade. Through the next century, even. So what will we do? Cling to our narrow notions of ourselves and get sicker and sicker? Or adapt our understanding of what we are, to come to see ourselves as a community of self? Now, as a futurist, I'm trained to look for where the future is just popping into existence today. And I think that the first signal that I saw of this emerging future was talking to Larry Smarr, director of Cal IT2 and Uber Quantified Self. Some years ago, we were talking about his regimen, and he said to me, "My body is an ecosystem." And at the time, I thought it was a metaphor, a really elegant metaphor. But it wasn't. He was speaking quite literally. Later, I started learning, as I'm sure many of you who listen to Science Friday have, about the human microbiome, about the trillions of species that inhabit every corner of our insides and outsides, that our digestion and immune function are dependent on these invisible helpers, that the microbes in our guts and on our skin influence our moods, our cravings, our behaviors, that this cloud of microbes extends beyond our bodies and is shared with households, with pets, even in big rooms like this one, that we're all getting closer together right now just by breathing on each other. The Human Microbiome Project, the American Gut Project, dozens of initiatives and thousands of scientific papers each year are building this new understanding. And the insights are very provocative, albeit couched in healthy scientific skepticism. But more and more biologists, Bonnie Basler, Jonathan Eisen, dozens more, are taking to stages like this one to educate and provoke. And they're giving us a new answer to that question of what are we? So what are we really? We are our germs. More than that, we are ecosystems, communities of human and microbial cells. And that realization has the power to change our assumptions, has the power to change the future of how we help and heal. It doesn't make sense anymore to wage a war on all germs, because we're just now beginning to understand the casualties. Like many of those drawn to this topic, I understand the implications through my own experience. When I was 14, I developed a mysterious rash. And the medical response was to slather my whole body in antimicrobial creams. Within weeks, the rash was gone, only to be replaced by dry, flaking lesions that cracked and bled. I stumped dozens of dermatologists before settling with the diagnosis atopic dermatitis. There's something wrong with your skin. Recent research suggests that the diversity of my skin microbiota may never have recovered from that early well-meaning treatment. There's even evidence linking it to the allergies and asthma and depression that I've experienced ever since. The point being, if we are ecosystems, we won't want to use these drugs in the same way. There's a lot of science to be done before we understand what better actions might be. Some microbiome researchers are experimenting on themselves, getting a dog, planting a garden. But we can take a leap out into this future if we approach the early science with openness and imagination. We can reimagine health through the shifts in our metaphors. Here are some big ones. Germs are bad. Becomes helpfulness and harmfulness is purely a matter of context. A helpful bacteria in the wrong place at the wrong time can still make you sick. Medicine as war gives way to gardening and wilderness stewardship. And finally, the self as an individual gives way to the realization that a person is a complex community of organisms, human and otherwise. In my day-to-day work, I take this mix of culture and science to tell stories about the coming decade. On my weekends, I'm writing a novel telling a story of this future 100 years from now. Let's meet in the middle. Let's call it 35 years. That's about a generation. Think of the small children you know, and think about them growing up. Are you ready? The Doctor Who fans in the room will know that we just time traveled. Welcome to 2049. And you know, time traveling always makes me hungry. So walk with me. We're going to the farmer's market. On the way, we're steered through a part of the neighborhood that I don't normally walk through. I'm wearing some invisible computer, and it keeps me from getting in a rut. When we arrive, we snack on some samples, make some purchases. You shake the farmer's hand, and we return back to the building where I live. What was once a parking lot is now a hydroponic garden. We pick the last of what we need for dinner and join families, prepping meals in the community kitchen. And we add our dish to a potluck with all the neighbors. And what a spread! There are 789 species of plants, animals, and insects in this meal. You remember we would have maybe 3 or 4 species on our plate? But today, there are perennial grains growing in the edible meadows up in the hills, and mealworms raised in a shack down the street, and more varieties of fruits and vegetables than I had seen in my life before 2020. I take out a self-cooled can of live cultures, suspended in water, and mist my plate. These are my happy bugs. They keep me emotionally balanced. And what a crowd! 4 generations, people of all walks of life. Those who could afford to live here a few decades ago, and those who frankly couldn't. Most importantly, healthy people live with sick people to take care of each other physically, but also microbially. It's like a sort of reverse quarantine, with the healthy influencing the sick for the better, and the sick keeping their neighbors' immune systems strong. It's a bright future here in 2049. Back in the present, it seems very bright, almost utopian. But it's simply imagining the possibilities that are open to us if we embrace this paradigm shift and resolve to use it to take care of ourselves and each other. Our alternatives are fairly dire. Our losing war against antibiotic-resistant microbes, the continual shrinking diversity of our diets and really all other life on the planet, and the shame and frustration of insisting that singular individuals take the blame for their poor health when our problems demand community. Taken all together, there are some aspirations that can help get us to such a healthy and caring future. We should strive for diversity over efficiency. And that goes for everything from our skin to the variety and equality of our global food system. We should strive for integration over segregation, because it's the differences between us, ethnic, cultural, in economics, in health status, that are the key to resilience in our communities. And finally, we should strive to reframe this individual notion of self, to better prepare ourselves for collective action so that we can meet these crises of the 21st century. It starts with us. Thank you.
All right, well, that brings us to the end of the lecture on the human microbiome. I hope it fascinates you as much as it fascinates me. And this is really helpful for integrating some of the things that we're learning throughout the semester and bringing them together in kind of a more visible whole. So please take some time to process what was presented in this presentation. Please write down any questions that you may have about the material, and also write down some questions that you think would be good test questions going forward. It's a great way to help you study for what's coming on your next exam. In the meantime, have a great day, and we'll see you soon. Bye.